Hello dears, welcome to SL Biologica channel, the foundation to life science. In this video, we are going to discuss some previous year questions from the topic of proteins. These questions are useful for your NEET biology preparation and various PhD level entrance exams like CSIR, ICMR, ICAR, DBT and GATE. And this is also useful for your state and central PSC level exams like BIOCHEMIST, Scientific Officer, Junior Medical Officer, Research Officer, etc. So first question is, which of the following is a correct characteristic of an alpha helix in a protein? Options: A. It has hydrogen bonding into or more running parallel chains. B. There is intra-chain hydrogen bonding in single helix. C. No hydrogen bonding is seen. D. It is a tertiary structure. So we have to find out the correct feature of an alpha helix in a protein. So we know that alpha helix is a secondary structure form of a protein. So it will have hydrogen bonding structure. But it has hydrogen bonding into or more running parallel chain. This feature is incorrect one because this is the characteristic of a beta plated sheet format where it exists in the form of sheet-like structure. So we can remove this option A. Then coming to option B, there is intra-chain hydrogen bonding in single helix. This is correct one because alpha helix is a coil-like structure format. So here we can see intra-chain hydrogen bonding, not inter-chain hydrogen bonding. Okay. So automatically we can remove this options like no hydrogen bonding is seen. That is incorrect. D. It is a tertiary structure. That is also incorrect one. So the answer is option B. There is an intra-chain hydrogen bonding in single helix. So this is the characteristic of an alpha helix of protein. Second question. So basically we have to discuss some important points of alpha helix here. So this is a structure of alpha helix where you can see intra-chain hydrogen bonding and it exists in the form of a rod-like structure. Okay, so intra-chain hydrogen bond stabilize the coil structure known as alpha helix. It's a rod-like structure. Example, alpha keratin, which is found in skin and its appendages such as hair, nails and feathers. So this is the overall protein structures. We know that protein exists in 4 types: primary structure, secondary structure, tertiary structure and quaternary structure. Primary structure means simple amino acid sequence. It exists in the form of a polypeptide chain. Where secondary structure is more advanced form of primary structure, where primary structure combined together to form a secondary structure. It exists in 2 forms, beta sheet and alpha helix. Okay, beta sheet we can see here that it exists in a sheet form. Okay, then alpha helix is a coil form. It's a single coil helix form. So alpha helix and beta sheet structures produced by hydrogen bonds forming within the polypeptide. In beta sheet we can see intra and inter-chain hydrogen bonding because it exists in the form of sheet. So bonding will take place inside and between outside the molecule. Okay. Then coming to tertiary structure, this is 3D overall fold of the protein containing secondary structure. So secondary structure again fold together to form a structure known as tertiary structure. This is a 3D form of a protein structure. Here we can see alpha helix structures, beta sheet structures, random coil. Okay, then coming to last structure type, quaternary structure. Here protein exists in the form of subunit. It is a multi-subunit complex structure where each subunit is represented as a polypeptide form. Here we can see 3 polypeptides, polypeptide 1, 2 and 3. So quaternary structure of protein is a multi-subunit complex where each subunit is a distinct polypeptide chain. Okay, then coming to second question. length of a alpha helical polypeptide chain of 20 residues spanning hydrophobic region of membrane would be. Here we have to find out the length of a alpha helical polypeptide, which consists of 20 residues. Okay, options: A 30 angstrom, angstrom meaning 10 raised to minus 10 meter. It is equivalent to 10 raised to minus 10 meter. 1 angstrom equivalent to 10 raised to minus 10 meter. So B 54 angstrom, C 5.4 angstrom, D 3.6 angstrom. So answer is 30 angstrom, 30. This is the formula to find out the pitch or length of the alpha helix. This is the constant unit of alpha helix forms. That is here translation distance of alpha helix structure is multiplied with the number of residues present per turn, that is 3.6, which results into 5.4 angstrom. Okay, so pitch of the alpha helix, that is length of the alpha helix, is equal to translation distance into number of residues per turn, that is 1.5 into 3.6 will give 5.4 angstrom. But in the given question, they have given 20 residues. So we have to replace this value 3.6 with 20. So 1.5 is a constant one. That is a translation distance of alpha helix. Were 20 residues. According to the given question, this alpha helical polypeptide contains 20 residues. So we have to multiply 1.5 with 20, which results into 30 angstrom, that is 30 angstrom. Okay. The next question: which one of the following is unfavorable for protein folding? Options: A hydrogen bonding, B van der Waals interaction, C conformational entropy, D peptide bonding. So here we have to find out the unfavorable thing for protein folding. We know that for the protein folding, we need a different type of bonding. So hydrogen bonding is needed one, van der Waals interaction. This is a basic type of interaction. This also needed. Then conformational entropy. This is not necessary one, or this is not needed. Conformational entropy. What do you mean by conformational entropy? Is it entropy associated with that conformation of protein? Entropy meaning degree of randomness of that particular structure or molecule. Here, conformational entropy is the loss of atoms' flexibility of movement and arrangement of a protein folds into its natural 3D structure is known as conformational entropy. So, conformational entropy is not needed for protein folding, so it is the unfavorable character for this protein folding. The option D, peptide bonding, is an essential bonding for this protein structure. So automatically the answer will be C, conformational entropy. So let's have a look about other interaction or other bonding, like hydrophobic interaction. Hydrophobic, from the name itself, it is water hating, opposite to hydrophilic. Hydrophilic meaning water loving. So hydrophobic interaction is shown by non-polar molecule when it interact with water molecules. So non-polar molecule have a greater inclination to cluster together in water than in other solvents that are less polar and exhibit lower self-association. This phenomenon is referred to as hydrophobic effect, while the interactions linked to it are known as hydrophobic interaction. So in picture we can see that this red indicates hydrophobic region, blue indicate hydrophilic region. So when this molecule or this isolated protein interact with water molecules, or when it dissolves in water, what happened? This hydrophilic region will interact with water molecule. Okay, hydrophobic region, that is non-polar molecules, will clump together towards inside. Okay. Then what is van der Waals interaction? Is a kind of interaction. It's a weak non-specific attraction force known as van der Waals interaction. It's produced when any two atoms come close to one another. Okay, when two atoms come close together, there develops a van der Waals interaction. It's a weakest one non-specific interaction. Okay. The basis of van der Waals interaction is that the distribution of electronic charge around an atom fluctuates with time. The distribution of electric charge surrounding an atom varies over time, which is the foundation of a van der Waals interaction. So basically, when an atom comes close together, they develop a interaction known as van der Waals interaction. The basis of this van der Waals interaction is the distribution of electronic charge surrounding these atoms. So based on the distance between these atoms, it may vary. Okay. Then coming to hydrogen bonding, we know that hydrogen bonding is a kind of strong interaction you can find in water molecules. This is the interaction between one electronegative atom with hydrogen atom. Okay. It is a chemical connection where the electronegative atom of one molecule is drawn to the hydrogen atom of another. So here you can see intrachain hydrogen bonding between two water molecules. Here electronegative atom oxygen interact with the hydrogen atom of another water molecule. So hydroxyl, sulfhydryl and amide groups participate in hydrogen bonding of protein structure. So molecule containing this hydroxyl, sulfhydryl, amide groups will generally participate in hydrogen bonding reaction. Next question. Glycerol is added to the protein solution to stabilize the preparation by A. Increasing the viscosity of solution, B. Stabilizing the pH, C. Preferential hydration of proteins, D. Interacting and neutralizing the surface charge on the proteins. So this is the structure of glycerol, CH2OH, CHOH, CH2OH. So what is the purpose of adding glycerol to protein solution? How it stabilizes that preparation? Answer is C. Preferential hydration of protein. So co-solvents like glycerol are frequently used to increase the stability of proteins in aqueous solution. Glycerol causes a native protein group to move into more compact forms, and when numerous proteins reform, glycerol also prevents protein aggregation. So basically it will hydrate that protein molecule and it causes that native protein to move into more compact forms. And next question is fifth one. Which bond helps in the stabilization of alpha helix coiled structure formation? We already discussed about this fact in previous question. Options: A. Glycosidic bond, B. Ionic bond, C. Peptide bond, D. Hydrogen bonding. So basically alpha helix is a secondary structure form of protein. In that structure we can see hydrogen bonding. So hydrogen bonding helps in the stabilization of alpha helix coil structure formation. Next question. Which of the following amino acids will participate in hydrogen bonding? Options: A. Valine, B. Glycine, C. Serine, D. Tyrosine, E. Glutamine. So here we have the combination of amino acids. Option A consists of A, B, C, B, B, C, D, C, C, D, E, D, A, D, E. So we have to find out which amino acids will participate in hydrogen bonding. So basically hydrogen bonding takes place between an electronegative atom and hydrogen atom of water molecule. So amino acids containing hydroxyl amino amide group will participate in hydrogen bonding. So it will be serine, it contains OH group, tyrosine again containing OH group or hydroxyl group, glutamine containing amino group. So option C, D, E, that is option C, which consists of combination of C, D, E. So amino acids with free hydroxyl, amino, amide and carboxyl group can participate in hydrogen bond. So here we have the structures of amino acid, valine. So this is the alkyl group of valine. It does not possess any hydroxyl, amino, amide, carboxyl groups. Again, glycine, this is the simplest form of amino acid where it contains hydrogen atom as alkyl group. Again, it will not participate in hydrogen bonding. Then serine contains OH group, that is hydroxyl group, so that it can participate in hydrogen bonding. Again, glutamine, it contains amino group. Then tyrosine, it also possesses a hydroxyl group. So these three amino acids will participate in hydrogen bonding. Next question. Amino acids are coded by dash number of codons. A60, B61, C62, D63. So basically we have 64 codons and we have 20 amino acids. Among 64, 3 will code for stop codons. So rest of the codons is 61. So option B, 61. Out of 64 codons, UAA, UAG and UGA code for stop codons. And rest of the 61 code for 20 amino acid. So amino acids are coded by 61 number of codons. Coming to next question, 8.1. Histidine protein buffer works at physiological pH of. If you know the physiological pH, you can directly answer this question. That is option C, 7. So this is the structure of histidine group. It contains imidazole group. This imidazole group side chain pK is nearly equal to physiological pH. pK indicates the acid strength. So this pK value is nearly equal to physiological pH, that is pH 7. So histidine protein buffer works at physiological pH of 7. Next question. Which structure of protein remains the same even after a denaturation process? A, primary structure, B, primary structure and secondary structure, C, secondary structure and tertiary structure, D, quaternary structure. Option A, primary structure. So we know that primary structure is the basic structural format of protein. It consists of amino acid sequence. So secondary structure and tertiary structure and quaternary structure will undergo some changes after denaturation process, but primary structure remains intact. Okay. So this is the primary protein structure. It is a sequence of chain of amino acids. The next question. Consider the structureless oligopeptide RGPS. This all letters indicate each amino acid. RGPS PKMPEY GSTDQSNWHFR. So this is a oligopeptide sequence. The number of bonds that will be cleaved by trypsin and chymotrypsin treatments separately are. So we have to find out the number of bonds cleaved by trypsin and chymotrypsin in this oligopeptide. So what is the function of trypsin? How it will cleave in this oligopeptide? Trypsin will cleave at C-terminal end of oligopeptide or amino acid chain. It cleave at lysine and arginine and enter C-terminal side. So here we have K and R, but it can't cleave at first and last amino acid side, and also this cleavable amino acid should not be surrounded by proline amino acid. This is the criteria for cleavage by trypsin enzyme. So it will cleave here it can cleave here. So K and R. So trypsin will cleave here at this position. Okay, so number of bonds cleaved by trypsin is 1. Okay, so we can remove option B and C. The next enzyme, chymotrypsin, it will cleave at the C-terminal end of aromatic amino acid. Okay, that is WYF, tryptophan tyrosine phenylalanine. Again, it should not cleave at first and last end of oligopeptide, and it will not cleave in amino acid position if it is surrounded by proline. So from here, it will cleave here at tryptophan C-terminal end, then it will cleave here at tyrosine position, because Y and W indicate tryptophan amino acid. This is an aromatic amino acid. Y indicate tyrosine amino acid, so it is also an aromatic amino acid. So 1 and 2, chymotrypsin will cleave at 2 bonds. So answer is option 1 and 2. So trypsin cleaves at C-terminal end of lysine and arginine, but adjacent amino acids should not be proline, will not cut at first and last amino acids. Chymotrypsin cleaves at C-terminal side of aromatic amino acid, but adjacent amino acids should not be proline and will not cut at first and last amino acids. Okay. Then what are aromatic amino acids? Phenylalanine, F, tyrosine, Y, tryptophan, W. And coming to the next question, which amino acid has more conformation available in Ramachandran plot? A. Proline, B. Glycine, C. Alanine. Answer is option B. Glycine, because glycine is the most simplest form of amino acid. It contains hydrogen as an alkyl group, so it will show less steric interference. This is because it just has a hydrogen atom as a side chain, meaning that it has less steric barrier. As a result, glycine can fill a larger portion of the plot, including areas that other amino acids are frequently prohibited from using. Okay. So what do you mean by Ramachandran plot? This is the Ramachandran plot where it depicts secondary structure forms of protein, and x-axis contains psi degree, phi degree, y-axis contains psi degree. It indicates C alpha C and C alpha N bonding characters. Okay, protein structures can be verified using Ramachandran plot, a graphical depiction of dihedral angles phi and psi of amino acid residues. Okay. Thank you for listening. Thank you.